Hello everyone, a warm welcome to this webinar on the topic of availability and use of standards in vaccine development. My name is Clive Nyaribo, project for the Vaccine Workforce Development Program. I work closely with the project co-leads, Professor George Warimwe and Marianne Munene. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share our questions during the presentation, and we'll address them during the Q&A session. We'll have a few open slots for the attendees to directly ask questions to the presenters. If your internet is stable, you can raise your hand using the available icon. Please take note that AI note takers and automated transcription tools are not permitted in this session. Unauthorized ports will be removed. This is a series of planned webinars with the goal of providing information on various topics in the field of vaccinology. This being run under the KEMRI-Wellcome Research Program, which is a world-known health research unit of excellence. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision of the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. The Vaccine Workforce Development Program is funded by the Gates Foundation and aims to expand the technical know-how and capability for sustainable end-to-end vaccine R&D and manufacturing in Africa through training and knowledge exchange activities in the vaccine ecosystem through a multidisciplinary and partnership approach. Reason why this is important is that Africa currently relies on vaccine imports and vaccine donations for up to 99% of its routine immunization needs. The Vaccine Workforce Development Program is investing in local talent and strengthening technical expertise, thus building manufacturing capability to improve vaccine access and reduce dependence. The Vaccine Workforce Development Program has 4 core initiatives. One, we have the Vaccinology School Engagement Program, where students from local secondary schools are engaged during open days and lab tours with career talks to spark their interest in vaccinology and careers within this field. 2, we have the vaccinology webinars like this one, where career scientists, graduate and undergraduate university students across the globe join webinars that provide information on various topics in the field of vaccinology. 3, we have the Vaccinology in Africa course, which is an annual master's level course that attracts participants from across the continent for strengthening theoretical knowledge on vaccinology. 4, we have the biomanufacturing apprenticeships that provides hands-on training in vaccine manufacturing facilities across the globe. Today, we are glad to have Dr. Selorm Avumegah, who will be discussing the topic availability and use of standards in vaccine development. Dr. Selorm is a global health expert with more than 15 years of experience helping to advance vaccines and solutions for infectious diseases from research to earlier clinical development. He currently serves as the program lead for the Biospecimen Sourcing Initiative and as a preclinical expert at the Coalition for Epidemic Preparedness Innovations, CEPI, where he supports global efforts to prepare for epidemic and pandemic threats. His work focuses on improving access to biospecimen, strengthening data stewardship, and supporting faster, fairer responses to global health emergencies. Before joining CEPI, Dr. Avumegah was part of the vaccine development team at the University of Queensland in Australia, where he contributed to the design of new vaccines for SARS-CoV-2 and other viral diseases. He began his career in Ghana, leading community-based health programs focused on sexually transmitted infections, neglected tropical diseases, and reproductive health education. That area of work continues to shape his commitment to practical, people-centered global health solutions. He is a trusted voice on vaccines, epidemic preparedness, and equitable access to health innovation, and regularly contributes to conversations that help shape global health priorities. Dr. Avumegah holds a doctorate in infectious disease and medical microbiology from Deakin University and has trained in global health policy. He is passionate about mentoring the next generation of scientists across Africa and about connecting science, policy, and practice in ways that create real-world impact. Outside his professional work, he is also a hobbyist poet. Over to you, Selorm. Thanks. Dr. Selorm, you're on mute.
Yes, thank you, Clive, for inviting me for today's webinar, and I'm very excited to be here. I will go off camera just so that we can concentrate and not get distracted. So good morning or good afternoon, everyone, depending on where you are joining from. Thank you for being here I'm really pleased to present this webinar on the availability and use of reference standards in vaccine development. Over the next 45 minutes or so, I would like to unpack why reference standards matter so much, even though they often sit quietly in the background of vaccine R&D. You see, reference standards may not be the most visible part of vaccine development, but they are absolutely essential. They help us measure potency and immune responses in a consistent way across labs, countries, and vaccine platforms. And COVID-19 made this very clear. Without common benchmark, it becomes very difficult to compare results or make confident decisions quickly. So by the end of this session, I hope you will have a clear, practical sense of what reference standards are, why they are needed, how they are used across vaccine development, and what is being done to make them more available for emerging pathogens. So just to set the scene, I will start this with a simple question. Why do vaccines matter so much in the first place? So you see, vaccines are it is said that vaccines are an excellent public health innovations because they proactively prevent infectious diseases at a population scale rather than treating them after people get sick. And this is believed to rank second only to clean water in reducing global disease burdens. So if vaccines are this important, the next question is how do we make sure they are measured, compared, and trusted consistently across different settings? So this will be the outline for my presentation today. I'll talk about why reference standards were needed in the first place and why they are so important for vaccine development today. Then I will walk you through the different types of reference standards and who is responsible for developing, approving, and maintaining them at the global level. After that, I will show how reference standards are used across the vaccine life cycle from early lab work and assay development through manufacturing and quality control, and even after a vaccine has been licensed after that. So I will spend some time also on the challenges we face, especially when a new pathogen emerges and we need standards quickly. And I will end with a short example of how CEPI is trying to address this through the Biospecimen sourcing initiative. So let's start with a bit of a history because the original problem is still very relevant today. So let's go back to the early 20th century for a moment. Doctors were using diphtheria anti-serum to treat patients, but there was a major problem. One batch could be very potent, while another could be much weaker. There was no common unit for measuring potency at that time, so people could not be sure that a dose meant the same thing from one producer to another. So Paul Ehrlich, one of the pioneers of immunology, saw this problem very clearly. Some children recovered after treatment, while others did not. Not necessarily because the treatment idea was wrong, but because the strength of different batches varied. So the central question became, how do we make sure a biological product has the same effect wherever it is made or used? The answer was to start treating biological problem a bit like weights and measure. Just as we need a shared definition of a kilogram or a meter, scientists needed a shared way to define biological potency. So what did they do? They introduced the idea of reference standard. So the solution was quite simple but quite elegant. Scientists created a well-characterized gold standard batch of a biological product, such as an antitoxin or a vaccine, and assigned it a defined number of units. Then whenever a new batch was made, they could test it against the reference and ask, does this new batch behave the same way? If the new batch produce only half the biological effect, it could be assigned half the biological activity. And if the batch, if it matched the reference standard, it could be assigned the same biological activity. This gave scientists and regulators a common language for potency, even before they could fully measure all the underlying molecules. So the basic idea, comparing an unknown sample to a trusted standard, is still at the heart of vaccine standardization today. But what is the relevance of this so much to vaccines? This matters especially for vaccines because vaccines are complex biological products. With a simple chemical medicine, measuring the amount of active ingredients may tell you a lot. But with vaccines, the key question is not just how much material is there, it is, does it produce the right biological effect? Okay, so that is why mass alone, micrograms of protein, for example, is not enough. Two vaccines could contain the same amount of antigens, but one may trigger a much stronger immune response than the other. So a reference standard help us measure the functional activity in a consistent way. So if a vaccine batch is tested against a reference standard, we can say something meaningful about its potency. And if different labs are using standards linked to the same global reference, their results become much easier to compare. So let me just take a step back and summarize the core principle that make this system work effectively. So a vaccine potency measurement rests on three key pillars. First, we need a good assay, a test that can measure the biological effect we care about. The second is that we need good statistics so we can interpret the assay properly. And third, we need a stable reference material, the anchor that everything else is compared against. So if any one of these pieces is missing, the system becomes much weaker. Okay, so, but the reference material is essential, is especially important because it gives everyone a shared yardstick to work with. Without it, different labs may still generate numbers, but those numbers may not mean the same thing. And we can see this during the COVID-19 pandemic. So just to put a bit of things in context, because I worked also on the COVID vaccine whilst in Australia. So COVID-19 really gave us a very clear, real-world example of why reference standards matter. At the start of the pandemic, vaccine development moved incredibly quickly. More than 200 vaccine candidates were being developed around the world using different technologies, different assays, different reporting units. The problem was that early on, there was no common biological ruler that everyone could use. One study might report antibody titers in one way, another might report binding antibody level in another way, and another might use a completely different assay. So when people try to compare immune responses across vaccine candidates, it was often like comparing apples and oranges. So the lesson was very clear. If we want to compare data quickly and confidently during an outbreak, we need a common standard much earlier. So COVID-19 was actually a wake-up call. It showed us that without a common yardstick from the beginning, it becomes much harder to compare vaccine candidates, interpret immune response, or make fast decisions. WHO and key laboratories such as NIBSE did develop international standards during the pandemic, including standards for measuring COVID-19 antibody levels. But many trials were already underway by then. Okay, so if those standards had been available earlier, developers could have reported results in a common unit much sooner. This is one reason initiatives like CEPI's 100-day mission places so much emphasis on having a reference standard and assays ready as early as possible. The goal is simple, right? When the next outbreak happens, we want scientists around the world to be able to speak the same measurement language from day one. Okay, so now that that is the idea of this of the need for the reference standard. But just let me give you a bit of information as to the bodies that really make this happen. So we have broad quality framework such as ISO and ICH, which are very important for quality systems, manufacturing, and regulatory harmonizations. But those frameworks do not usually tell us exactly how to measure the potency of specific vaccines. That is where the WHO Expert Committee on Biological Standardization, or ECBS, come in. ECBS focuses specifically on biological products, including vaccines. It helps establish the physical reference standards and guidance that allow different labs and regulators to measure biological activities in a consistent way. So I will let you know how closely ECBS and the WHO reference standard really interplay and work together. So you can think of the ECBS as the global decision-making body for the biological standards. Experts and regulators review the evidence from collaborative study and decide whether a candidate material should become an official international standard. Once adopted, that materials becomes the global benchmark for use. So, for example, an Ebola antibody preparation could be formally assigned a value in international unit and then used around the world to calibrate antibody assays for Ebola vaccine development. ECBS also produces written guidance, so it does not only provide the physical tools, it also helps define how tools should be used so that in that sense, it brings together the material themselves and the rules that help the global community measure biological activity. So with all of that information in mind, what do we mean by a reference standard? So think of a reference standard like a gold standard ruler in a lab. It is precisely measured and carefully documented, and everyone uses it to make sure their own measurement line up correctly. So put simply, a reference standard is a trusted, well-characterized sample that serves as a benchmark to check the quality, identity, or potency of other samples. By comparing tests and materials to this standard, we ensure results are accurate, consistent, and meet the required specification. So that is why this is so important. But let me just also bring a bit of a difference between these two. So this slide, as you can see, is drawing a distinction between two types of standard, an antibody standard and a vaccine standard. Antibody standard helps us measure the immune response to a vaccine. A vaccine standard helps us measure the vaccine product itself. So antibody standards are usually made from serum or plasma containing antibodies. They are used to calibrate immunogenicity assays so that different labs can express immune response in a comparable unit. One major advantage is that a single antibody standard can often support many vaccine candidates targeting the same pathogen. However, vaccine standards, on the other hand, are reference batches of the vaccine product itself. They are mainly used in manufacturing and quality control to check whether a new vaccine lot meets potency specification. So the key point is that vaccine standards are usually product-specific. Vaccine standards are product-specific, while antibody standard can sometimes be more broadly used across different vaccine platforms. That distinction became very clear during COVID-19, when many different vaccine technologies were developed and available. Okay. So this slide looks a bit technical, so I will take my time and break it down just for clarity. When we talk about antibody standards or antibody assays reference standard, a reference standard support three related activities: assay calibration, assay validation, and assay evaluation. So when we say calibration, calibration is about setting the measuring scale. The WHO international standards sit at the top of the scale. Secondary, tertiary, and working standards are then calibrated against it. So day-to-day testing remains linked back to the global gold standard, reference standard. Validation asks a slightly different question. Does the assay actually work for its intended purpose? So here, standard help assays perform well across, a standard helps define the scale in validation, while sample panels help test whether the assay perform well across low, medium, and high immune responses. Evaluation is about comparing results across labs or studies. So if everyone calibrate to the same international standard, then results from different places become much easier to interpret together. So the key message is that in simple terms, calibration builds the scale, validation checks the assay, and evaluation gives us confidence that results are comparable. I believe that I've mentioned several times about in the same unit, international units, right? So so what is international unit? And put simply, IU or international unit is a standard way of expressing biological activity. It is not just a weight of concentration. It reflects a biological effect. I would like to describe IUs as the common currency of biological activity. If every lab report results in its own local unit, comparison of becomes very difficult. But if results are converted into international units, it becomes much easier to compare data across studies, countries, and vaccine candidates. Okay, and this is especially important for vaccines and biologics because the amount of material present in a vial does not always tell us how active it is. The products may contain similar amount of antigen, but their biological activity may differ. And so IU help capture that functional potency in that regard. So how is international antibody standard established? So to establish a new WHO international standard, scientists first identify a candidate material. That material is then tested through an international collaborative study where multiple laboratories examine how it performs in different assays and settings. The results are then reviewed by ECBS, which is the Expert Committee on Biological Standardization. If the evidence shows that the material is suitable and stable, the ECBS can formally adopt it as a WHO standard and assign it a value in international unit. Okay, so that is how this is done. And when it comes to the life cycle of this standard, WHO standards usually do not have a fixed expiry date in the way many medicines do. They are designed to be very stable, often freeze-dried and stored under carefully controlled conditions. As long as ongoing monitoring shows that the material remains stable, it can continue to be used. And some standards have remained valid for many years, even decades. Okay, so eventually, if of course a stock runs low, and when that happens, a replacement standard is produced. So now let's see how this global standard connects to the day-to-day work of a local laboratory. So let's think of traceability and linking up this standard. So traceability is the link between the global standard and the result produced in an individual lab. Imagine the WHO international standard defines what 100 international unit means for a particular antibody. So 100 international unit. A national lab may calibrate its own national standard against that WHO standard, and then a manufacturer may then calibrate its working standard against the national one. So each step passes the value down the chain, so that when a local lab reports a vaccine batch or antibody standards in IU, that result can be traced back to the original WHO international standard. This is what allows results from different countries and laboratories to be meaningfully compared. Without traceability, one lab's number may not mean the same thing as another lab's number. Okay, so that is why international standard really ensures traceability, and traceability is very important for us understanding the vaccine potency and biological activity. So let's look at how reference standards are used, right, in data. So in early vaccine research and development, one of the first things developers need is a reliable assay. They may need to measure antigen content, neutralizing antibodies, or another immune marker. If a relevant reference standard is already available, using it from the start is a major advantage. It helps developers understand their assay's accuracy, range, and consistency. It also means that if several groups are working on vaccine for the same pathogen, their results can be compared on a shared scale. So at CEPI, this is especially important because we may support multiple vaccine developers working on the same disease. And if they all use common reference reagents, it becomes much easier to compare early clinical data and make evidence-based decisions about which candidate should move forward. Okay, the same idea becomes important again when a vaccine moves from the lab into the development process. So the reference standard is also used in process development and characterization. As vaccine production scales up, developers often need to adjust or optimize the manufacturing process. The question then becomes, has the product stayed comparable, or has the change affected the potency? So a reference standard here helps answer that question. Developers can test a batch made before the change and a batch made after the change against the same reference standard. If both show similar activity, that provides confidence that the process change has not reduced the potency. And this is also important for regulators, right? A reference standard gives everyone a common anchor for judging whether a process change has had a meaningful impact. So standard helps define product specification. For example, if a certain level of activity is needed for protection, then each lot can be required to meet that level before release. And this also brings us to the next level of use in quality control and lot release. In routine quality control, every vaccine batch needs to be tested before it is released. And a manufacturer will usually test the new lot against an in-house reference standard that allows the team to confirm that the batch falls within the expected potency range. In many countries, right, national control laboratories also perform independent lot release testing. If both the manufacturer and the national labs are using reference standards traceable to the same WHO reference, their results are much easier to compare. This helps reduce confusion, avoid unnecessary delays, and build confidence that the vaccine lot meets the required quality standard. Reference so that is a bit of that. However, reference standards also remain useful even after vaccine is even licensed. After licensure, reference standards continue to support vaccine quality and public health monitoring. For example, they can be used to compare products from different manufacturers to check that vaccine load continue to meet expected potency threshold over time. Okay, and they are also important in immunological surveillance. If different labs measure antibody responses using assays calibrated to the same international standard, we can compare population level immunity across studies or countries much more reliably. So reference standards are not only useful for development, they support the entire vaccine life cycle from early R&D through long-term monitoring after vaccine deployment. So this system only works because many groups contribute to it. WHO and ECBS provide the global framework and formal approval of standard. WHO collaborating centers such as NIBSC and PEI do much of the practical work of preparing, characterizing, storing, and distributing the physical material. National regulators and national control labs then use these standards in guidance, quality control, and lot release. Manufacturers use them in their own development and production processes. Regional networks and partnerships are also important because they help expand access, build capacity, and make sure these standards are not only available to a few well-resourced settings, but to everyone as a whole. So these are some of the things that I wanted to highlight today. So just by way of a quick summary or takeaway, the main message is that reference standards are fundamental to vaccine development, even if they are not always visible. They allow us to measure potency and immune responses in a way that is meaningful, consistent, and comparable. They also are important in vaccine science. They let you know that vaccine science is deeply collaborative. Standards are created and maintained through the combined efforts of scientists, regulators, manufacturers, WHO collaborating centers, and many other partners, right? So that is why this is very important, and it's a global effort. If we build reference standard into the vaccine life cycle from the beginning, we can make development faster, make data easier to compare, and support confidence in vaccine quality and performance. So before I finish today's talk, I want to share one practical example of how CEPI is trying to address some of the challenges that we have encountered in reference standard development or support through the Biospecimen Sourcing Initiative, which is a new project that CEPI has launched. So just a bit of information about CEPI. Some of you may already know CEPI, but for those who do not, let me give you a brief introduction. CEPI is the Coalition for Epidemic Preparedness Innovation. It was created in 2017 after the West Africa Ebola epidemic, when the world recognized the need for a more coordinated approach to developing vaccines against epidemic threat. And CEPI is a public-private partnership that brings together government, industry, philanthropic organizations, civil society, and scientific partners, and with the aim to accelerate the development of vaccines and other biologic countermeasures for epidemic and pandemic threat. CEPI's vision is to see a world in which epidemic and pandemic are no longer a threat to humanity. And a key part of that mission is making sure that vaccines and countermeasures are accessible to people everywhere. So CEPI has been supporting the development of reference standard as far back as 2018. And that was when we launched one of our requests for proposal seeking convalescent Lassa fever serum to support this effort. And through several partnerships and global engagement, there has been a lot of development in this regard. So this slide is very busy, but it illustrates the current status of international antibody standard for CEPI priority pathogens and viral families. It's a lot detailed, but it essentially answers which diseases already have reference standards and which do not yet. Our priority pathogens are grounded by viral families like arenaviruses, coronaviruses, and etc. And etc. But for each pathogen, there are columns indicating whether an antibody standard exists, whether an antigen standard exists, and a catalog number available from the NIBSC website that you can order from. So that is what this slide is trying to show. So just to sum up this, as of now, we have more than 15 new reference standard produced with strong coverage for antibody standard, but a significant gap also remains in antigen standard, right? So moving forward, our top priority will be to see how best we can fill this gap and move the science forward. As I said in the beginning, there has been several collaborations. This is through a several collaboration effort, and so CEPI has been working with different partners to make this happen. I want to just let you know that for vaccine development or WHO antibody standards, WHO antibody standards are preferably developed from individuals that have recovered from a disease. Okay. Because we believe that their blood or serum samples might contain important information that can help us develop the standard to support a vaccine development. And when you are sourcing for these materials, there have been so many practical challenges in sourcing for materials to support the development of reference standard. We have seen that we often have challenges with identifying survivors, building local partnerships, delays in ethical approvals, securing consent, and arranging transfer agreement in time. So in practice, these steps can actually take weeks, months, or even years, or even longer, because sometimes when we are doing all of this in an outbreak, it can take a very long time and the response cannot be very fast. It's not very fast. So the problem is very clear. During an outbreak, the traditional approach may be too slow. By the time a reference standard is ready, vaccine development may already be well underway. That is exactly the gap that the Biospecimen Sourcing Initiative is trying to help address. So the BSI is about being prepared. It's about pre-positioning the capacity to collect outbreak specimen quickly, ethically, and in a way that support vaccine R&D. So if a new outbreak occurs, the aim is to have trusted partners ready to collect and help generate reference standards and assays needed for faster response. So that is what the BSI is about. And a bit of the project overview of the BSI. We are rolling this out in 4 phases to ensure and support its operationalization for the next pandemic. Phase 1 has is by establishing governance management framework and all necessary documentation for the initiative. Phase 2 involves launching a call for proposal to recruit BSI partners. And we plan to really select about 20 geographically diverse partners with strong capabilities in outbreak sample collection and reference standard development. And phase 1 and phase 2 has already been approved and they are all underway. Phase 3 is when the rubber meets the road, actually really collecting a biospecimen and developing reference standard through the new partnership in response to an outbreak. Finally, we have the phase 4, which covers pressure testing the system with simulation exercises and maintaining the network so that it is ready for a real disease scenario. So that is what CEPI is trying to support, do to support this ecosystem of reference standard development. So how would this actually play out? So if BSI were fully operational during an outbreak, in an outbreak scenario, the BSI team will rapidly be activated to support collection of relevant materials to support this activity. And a key feature of the BSI is having a pre-negotiated agreement in place among partners. This means that when samples need to be shared across borders or sent to a specialized lab, the legal groundwork is already done to support this effort. So to that end, we have launched a call for proposal to recruit new partner institutions that have the proven track record in outbreak biospecimen collection, reference standard development, storage, or related areas. And the main outcome will be to establish a ready to activate BSI partnership network spanning multiple regions and multiple continents. The BSI call for proposal is currently open with the proposed deadline of 12th of August, as indicated on the slide. So if you or your organizations are interested, or if you have any suggestion, please direct them to our CEPI website. And there are so many references on this. The WHO reference standard, there is a ECBS website that all of this information can be found. I have the NIBSC website, and so all of this information are available, and I'm willing to provide more references if anyone needs that. So for more information, please do reach out, and let's keep the conversation going. So with that, thank you very much for listening, and I'm happy to take any questions or comments. Thank you.
Thank you, Selorm. Thank you, Selorm, for such an insightful talk on standards. There are a number of questions on the chat, but first I would like to understand in the current context of the Ebola outbreak. I understand you are setting up the PSI, but have you been able to correct maybe specimen to support the development of vaccines in the current context of the Ebola outbreak?
Yes, so thank you very much. For the current Ebola outbreak, CEPI proactively worked with our partners in Uganda, UVRI, and we were able to collect some specimen from the 2007-2008 outbreak of the Bundibugyo Ebola virus from those survivors. So we're able to work with UVRI to support that effort. And we are currently also working with several partners in DRC and also in Uganda to see how best we can also support and have access to samples relevant to develop the first Bundibugyo reference standard. So the collaboration is ongoing with various partners in DRC, INRB, and also partners in Uganda, specifically UVRI, to move this effort forward.
Thank you. There is a request from John Gitonga. Kindly elaborate on the third bill. could go back on the slides where you were there talking about some pillars.
Can you please repeat the question?
Yes, there is a question on the chat from John Kitonga saying, "Will you kindly elaborate on the third pillar?"
What? Oh, on the third pillar. So the third pillar was, if you look at the presentation, I talked about the three pillar, which was the quantitative assay, having a very good statistic to interpret the data, and the third pillar was on having a reference standard available to really put through the assays. So I don't know if you're referring to the reference standards or you're referring to the BSI pillar. So that's, I will need some clarification on that. Is it the BSI project overview pillar, or the person is referring to the reference standard pillar that I referred to? But I hope that answers the question.
Standard, yes. Yes. Also, there is a question from Pete Gardner. What are the ways to ensure your standards and assays are relevant prior to having vaccine efficacy or mechanistic correlates of protection? How much guesswork is involved?
So thank you very much. So I think that previously, many a times where you are developing a vaccines and you don't have the right standard, many people use animal generated material to develop the reference standards. So usually at the preclinical early development stages, people use different sources, animal, let's say plasma or serum standard derived from animal sources, mouse, NHP, rabbit. They use that for the reference standard development. But then when a WHO endorsed reference standard becomes available, it is encouraged and recommended that that lab kind of back calibrate it against that WHO international antibody standard. This will give you an idea of how your vaccine, this will support the validation of the assay, but it will also help support, to give you good information to know how your vaccine candidate is performing against a clinically relevant standard, right? So how to ensure that there are many animal sources for reference standard. However, to have it, you must calibrate it against the WHO gold standard when it becomes available to ensure your assay and your vaccine candidate is meeting the required global standard. I hope that answers that question. Yeah.
Thank you. There is a comment from Roslyn Kinoti saying that I have used the NIPSC SARS-CoV-2 international standard to benchmark an in-house secondary standard and put my pseudoviral neutralization assay. It worked really well and gave us confidence in our data output.
Perfect. No, thank you very much.
The importance of having those standards.
Yes.
There is a question on the Q&A from Sion Dembo. What measures are put in place to ensure ethical standards are followed in specimen correction?
What measures are put in place to ensure ethical standards are? Okay, so no, thank you very much. This is a very important question. So before we work on this, right, it is very important that we work with national institutions, global, especially national institutions. So because the national institutions have our partnership with them is very important to ensure that CEPI support to those institutions to access specimen to support reference standard. They are following their local, national regulations and guidelines. It's very important. So as part of our work at CEPI, it is part of the deliverables and requirement that every partner that we are working with that CEPI is supporting, let us know those approval mechanisms and show us evidence that this has been thoroughly reviewed by their in-country ethics board, institutional board, and following the country regulations and also following international norms. If they don't meet this, then there might not be a choice partner for us to work with, and we will work with an institution that can provide us all of these regulations and that they are following the right regulation. So that is how we ensure that ethics is achieved and we are not going against any national regulations and also international regulations. So this is a very stringent there's a very stringent process in place. It takes time. That is why it's very difficult to do this in an ad hoc outbreak setting. And so that's why the BSI, we are trying to pre-position all of this ahead of time so that we understand and work through the bottlenecks before the next pandemic. So Zion, there is a very strong ethical procedure in place to ensure that everything that we are doing is meeting the global recommendation and country regulations as well.
Thank you. There is a question from Darren asking about what standard is mostly used in Africa for GMP production of vaccines. WHO GMP, PICS guidelines, how closely is Annex 1 EU GMP followed currently?
So thank you very much. Then I don't have an off-the-head answer to that question, but I can reach out to some of my quality members, our quality department, and they will be able to give me more information, and I will provide this to Clive, to you. I don't have a quick answer to that.
Yes, thank you. But I suppose most of the NRAs in Africa, where vaccine manufacturing is in infancy, actually, they closely borrow from all the standards, whether it's WHO, technical series, PICS guidelines, and also the Annex 1 EU GMP. So they do follow those. So they borrow heavily from those standards developed by IMA, yes, and other international guidelines. Thank you.
Thank you, Clive.
Yes, there is an opportunity to ask a question directly to the speaker. So you can raise your hand and the speaker will answer your question. Yes, John Gitonga. John Gitonga, kindly go ahead.
Thanks for the nice presentation. I had a questions in regards to the vaccine immunogenicity assay validation. Could you please indicate which standards or guidelines or even regulatory references are used to define the acceptable criteria for parameters such as accuracy, linearity, and even range? Are there specific internationally recognized acceptance thresholds for these parameters? Thanks, and over.
Yeah, thank you very much, John. There is a guideline on that, and that is why I'm on this screen. I'm showing you one of the references that I use, which is the WHO Manual for the Preparation of Reference Standard for Use as Secondary Standard in Antibody Testing. This is a manual produced by WHO and has all of the information on those threshold acceptable guidelines for assay standardization and calibration and evaluation. So I will entreat that you because it's quite comprehensive, and so I will not be able to answer that because that is the whole assay calibration, validation, evaluation is quite a very robust activity. So I will like you to visit this website, the second one, the WHO Manual for the Preparation of Reference Material for Use as Secondary Standard. It will give you all of those indicators as to those acceptable threshold and the unit. So But this is a very important question, and that is really at the heart of today's presentation. So thank you very much.
Thank you, Selorm. Maybe I could just ask, adding to that, what is the role of maybe other pharmacopoeia like the USP and PP in developing these standards, maybe?
So no, thank you, Clive. So as I said, in the beginning or throughout within the presentation, I mentioned that there are so many global partners, right? We have several WHO collaborative centers, and we have several collaborative centers, WHO collaborative centers across the globe, right? And also when NIBSC is helping with the development of the standard, they also they reach out to several collaborative partners. So there are many partners that we leverage in the US and also in Latin America, in Africa, in Asia. So it's very important that the effort is very global and at different settings when establishing the reference standards. So just to say that that we do have partners in US and other places that support this effort.
Thank you. And closely related to equity, to ethical structures in place, how will you ensure there is access to the treatments and vaccines being developed? Because majorly they are being developed maybe outside Africa. Access to this. Yes.
Can you please repeat the question?
Yes, I'm asking, closely related to the ethical structures being in place, how do you ensure that the treatments are accessible to the participants, maybe the ones who even provide samples?
So no, thank you, Clive. So I just want to say that when the reference standards are developed, it's a global tool, right? It's a global tool and it's accessible to everybody on the globe. So in terms of equity, when reference standards are developed, it's anyone, any lab, any researcher, any scientist does have access to this material because it's part of the global. It's part of the WHO's support to the global community. So that is how equity is embedded into the reference standard development approaches.
Thank you for that clarification. Yes, there's an open slot for 2 more questions from the audience, then we close. Yes, if there is no another question, Dr. Selorm, thank you for collecting time for this. I would like you to make your closing remarks. Thanks.
Yes, thank you very much, Clive, and for inviting me for today's webinar. And thank you very much, all of you who were able to join. You've been a very good audience. And I will again encourage all of you to visit CEPI website and have a look at the BSI call for proposal and see whether you'll be interested or you have an institution in your country that will be interested. So thank you very much for inviting me and for listening. I appreciate the time. Thank you. Over to you, Clive.
Thank you, Selorm, and thank you very much to everyone in this webinar. We would like to hear about your experience and what else you would like us to discuss in upcoming forums. Before we close, kindly requesting that you take a few minutes to respond to the post-webinar survey posted in the chat. Have a nice day. Looking forward to more engagements with you. Thank you.